Hello, greetings from Vienna and welcome to my talk on fasting, fasting mimetics, microbiota and epigenetic regulation. Caloric restriction and fasting have been discussed for sustaining health and longevity since the antique. Modern molecular biology analyzing the hallmarks of aging have seen multiple effects of both fasting and caloric restriction on mechanisms such as epigenetic methylation, mitochondrial function, inflammation, DNA repair, telomeres, and many others. Interestingly, microbiota are not typically included in the figures of the hallmarks of aging. However, we should be clear that caloric restriction and fasting need to be differentiated in terms of timelines. Butyrate and beta-hydroxybutyrate, for example, need days for a significant increase in contrast to caloric restriction such as intermittent fasting. Soon also the effects of different diets such as caloric restriction or ketogenic diet on metabolic improvements have been attributed to changes in gut microbiota. And caloric restriction and fasting regulate epigenetic mechanisms, especially histone deacylases, sirtuins, via short-chain fatty acids, beta-hydroxybutyrate, and maybe other mechanisms. Unfortunately, caloric restriction and fasting seems to be something like a nightmare for many consumers, and so we started soon to look for pharmaceuticals or functional foods which mimic the main pathways of fasting, SIRT, AMPK, TOR pathway, and indeed we found substances like rapamycin and metformin, or functional foods like polyphenols, epigallocatechincolate, or spermidine. This started the hype of fasting mimetics on mechanisms such as autophagy, inflammation, senolysis, cognitive functions, and others. The highly different responses to diets and food ingredients have been discussed in the light of quite individual microbiota composition and cross-feeding, and obviously it's still quite difficult to predict individual responses to specific fibers. Additionally, different microbiota structure may also be responsible for the quite different metabolization of polyphenols. To explore similarities of fasting and fasting mimetics, we first characterized specific plant ingredients, polyphenols, and their effects in senescence and adipogenesis in pre-adipocytes. And we found that especially the green tea epigallocatechincolate, but also other polyphenols like anthocyans, resveratrol, and others stimulate SIRT3 expression and reduce senescence as indicated by beta-gal or the inflammatory phenotype such as IL-6. So with these experiences of different plant ingredients, polyphenols, we designed a study comparing fasting and fasting mimetic SIRT-FOOD shot with the ingredients we determined before in vitro. We got feces and blood spots in a group undergoing Buchinger fasting, which is below 120 calories per day for 7 days on the one hand side, and in a group of volunteers taking this SIRT-inducing fasting shot and this was going for 3 months, and we had 171 participants. We analyzed microbiota by Illumina sequencing. We did methylation of line 1 and expression of microRNAs and RNAs in the blood spots. Focusing on the first solid stool feces sample, after the Buchinger fasting, we found a rise in the distribution of proteobacteria, an increased microbiota diversity, and a significant increase in Christiansenella. Looking for interactions between microbiota and host metabolism, we found that the abundance of Christiansenella is inversely correlated with age and body mass. And interestingly also, we found a significant correlation between SIRT1 gene expression and Prevotella and Lactobacillus. Fasting indeed increased expression of SIRT1, SIRT3, microRNA LED7, which is quite important in the area of fasting and metabolism, and we also found a quite increased mtDNR. The 3 months intervention with the SIRT-inducing drink mainly increased the whole group of actinobacteria. Only fasting increased butyrate significantly. We found a slight increase in butyrate with the drink, and in general the Firmicutes Bacteroidetes ratio decreased and correlated significantly with the body mass index. In general, we found a decrease of VERRUCOMICROBIA with age, and quite interestingly, for microbiota host interactions, we found a positive correlation of the abundance of butyrate producing Bacteroidetes with telomere lengths. In conclusion, we found that fasting and to some extent also fasting mimetics, the SIRT-inducing drink, result in a beneficial modulation of microbiota, especially diversity, short-chain fatty acid, beta-hydroxybutyrate, and metabolism, specifically expression of SIRTs, mitochondrial DNA, telomere lengths. Of course, it would be important to get more information on follow-ups on the timelines after the end of the intervention. Interestingly, especially as a scientific point of view, the microbiota structure seems to interfere with the expression of sirtuins and also metabolism relevant microRNAs. Of course, we have here to look for the targets of the microRNA and their role in their interaction with microbiota. Thank you very much for your interest. I hope for an exciting discussion, and especially I hope to keep in touch with some of you. You can see here my email, my website, and especially my personal health website tries to focus on aspects of personalization, which are also, I think, in the center of interest in the area of microbiota. Bye-bye.